Hello everyone. So welcome to Mohit Agriculture tutorial or Mohit's Agrobiology, the new name that we have kept of this channel is. Right? So in this set of video, we are going to talk about insulin, right? What is insulin? How does it carries out the changes in the body? How does it signals all the sort of changes that are being carried out, right? Merely the main thing over here to understand is that insulin neither is an hormone nor is a sensory organelle, means a sort of sensory system. It's a kind of, you can say, polypeptide that has been created by the body to induce the appetite, right? Very firstly, primarily we will define the insulin, then we will go out to the pancreas, understand the production of the insulin, then we will understand how does this pancreas induce the insulin to carry out the appetite inducing system, right? The thing to understand here is that insulin, right? What is insulin, right? So primary thing is what is insulin. So merely when you're talking about insulin is a polypeptide that has been created by a special sort of gene that is in the beta cells of the pancreas. That is POMC set of genes. That gene synthesizes this sort of hormone by certain transcription process, and that gene is responsible for what all sort of things are happening in our body. If there is a glucose flux in the body, very primarily thing to understand is that this insulin is antagonistic to the glucose, right? Whensoever there is a glucose concentration in the body, this insulin will trigger a certain sort of sensory system to induce the glucose breakdown, or if not glucose breakdown, then conversion of glucose into its polymers, specifically fat in the adipose tissue, that's the white adipose tissue, or merely converting it into the glycogen, right? Now these are the sort of things that we need to understand primarily before understanding this insulin in detail. Now the thing that we understood is that insulin is antagonistic to glucose, that's okay. But what sort of changes does insulin carry out in our body, and what is the primary focus? Our primary focus here is going to be what sort of changes that's happen in the body when the insulin is produced in the body, right? The primary thing is here that insulin is being produced from the pancreas. Do not go for this spelling, right? So insulin is being produced from the pancreas. These pancreas are the signaling, you can say a sort of organelle that signals the production of insulin. But insulin is not solely produced due to this pancreas. Insulin is produced due to the beta cell of pancreas, right? Beta cell of what? Pancreas. Okay, so let's very firstly understand. Now let's first understand the structure of the pancreas. Pancreas has three sort of cells, right? It is the foremost organ in our body that carries out the channels to just demold that glucose or insulin in the body, right? Now it has three sort of signals, or three sort of cells that create different sort of hormones. Primarily is alpha cell. Alpha cell is responsible for the production of the glucagon. This alpha cell have particular gene that produces the glucagon, and the signaling criteria from the insulin is primarily very different, right? Beta cell, the one we primarily know as the insulin generating cells in the pancreas, are those sort of cells that create out signaling to induce the production of the insulin. Third primary cell over here is gamma cell, right? Gamma, or you can say it as a delta cell. So these delta cells are those sort of cells that are primarily focused in production of somatostatin, right? Somatostatin. Now these are the sort of primary things to understand before going into the basic detail of the topic. Now, what's the topic is saying, what we're gonna study here is that the production of the insulin. So the primary thing is to rub all these things and blank board over here so that we can understand the process, the signaling process that induce those POMC genes sitting inside the nucleus in the beta cell to be induced, to be transcribed, to be translated, to generate a sort of chain of polypeptide that is carrying the signaling for the insulin, that is further modified into its micro units to carry out the signaling whensoever there is a flux of glucose in the body. So let's go for it. Okay, so now our primary focus here is to understand the principle of the signaling that carries out in the beta cell to create the signaling cascade. So what I have made over here is a sort of cell, right? These are the beta cell. Beta cell in the pancreas contains a nucleus as usual, as every cell contains, and that nucleus has certain set of genes that have been essential for the production of these insulin molecules, right? Apart from that, the system of the endoplasmic reticulum system, that is the transport system, is very highly modified over here to carry out the changes that are being carried out during the translation or after the translation into the cell to carry out certain polypeptide to the downstream cell, right? Now, here we have made the three logic gates, right? These are the three gates that are essential for the signaling of this, this insulin formation, right? Now, the primary gate we have made here is GLUT4. GLUT4 is a gate that is really very essential for the production of the insulin in a beta cell, since beta cell contains a set of genes that we call as the POMC set of gene, POMC alpha, that has the signaling pattern, or that has a quite nucleotide pattern that will induce the production of the polypeptide that's necessary for the production of the signal, insulin formation signal, right? Afterwards, we will understand the sort of changes that have been took out into this insulin molecule from pre-proinsulin to the proinsulin to the insulin, where it is in the activated form is being generated to just carry out or induce the glucose molecule flux outside the cell or in the bloodstream, right? So what I have talked about is GLUT4. This one channel is the K+ channel, that's very simple, that's potassium. And this one is the calcium signal, Ca2+ signal. Now the changes that are being carried out into the cell are due to the flux that is being carried out over here in the bloodstream. Through the bloodstream, the signaling has went to this sort of cell, that is the beta cell, that the flux of glucose is higher outside. Once we know that the flux of glucose is higher the outside, this GLUT4 is an activated channel that carries out the signaling cascade and carries out the glucose molecules inside the pancreas, the beta cells, and the beta cell functions to just notice that the flux is higher of the glucose. How? Now this process goes through glycolysis, right? This process goes through the glycolysis, production of pyruvate. Pyruvate will go to citric acid cycle, and this citric acid cycle will be further carried out downstream to the production of ATP to further oxidative phosphorylation. Once oxidative phosphorylation have to place in this beta cell, the ATP flux will induce this K+ channel, the potassium channels, to close, to shut down, because these are the only channels that carries out the signal that there is no such flux of glucose outside the cell, since there is no flux of ATP inside the pancreas. Once this ATP is induced in the pancreas, this channel will particularly shut down and due to this channel getting shut down, this Ca2 plus channel, that's a calcium inducing channel, the flux of calcium inside the cell will increase, means this channel will just go activated, right? Now once this channel is activated, it means the flux of Ca2 plus is higher inside the cell, right? Primarily, the thing is that you should revise whatsoever I am saying. I am saying that the one that you are seeing are the 3 gates, and there is a channeling signal that carries out the signal to the ATP molecules to the K plus channel, and from the K plus channel the signal is being translated to the Ca2 plus channel or transferred to the Ca2 plus channel that induces the calcium flux, higher calcium flux inside beta cells. Once this higher flux is being activated, then further the protein, particular protein that binds to the Ca2 plus molecule, maybe any of the protein, I'm not going to just take the name over here or the receptor, that will go and switch on the transcription factors in the nucleus. Once these transcription factors are being switched on, that will induce this POMC alpha set of gene to carry out the signaling and to carry out the polypeptide to the mRNA. mRNA would be carried out to the endoplasmic reticulum, and endoplasmic reticulum over here would be translated into the polypeptide chain. That chain is inactive insulin, not active, because we know that in the endoplasmic reticulum and further in the this Golgi apparatus, further several processes are carried out. The processes are really very essential and very you can say needy for the cell, that to carry out any of the changes in glucose concentration or anything that changes has to be taken place inside the cell, right? Thence we are going to talk about those processes further, but very primarily again we need to focus on the signaling cascade. Again, a quick revision or quick recap is that this glucose signal channel is induced by the glucose flux outside in the bloodstream that is carried to the pancreas. In the pancreas, the glycolysis citric acid cycle induces the production of ATP. Once ATP flux is induced or increased inside the cell, then the K plus channel, which primarily shut down, and the activated Ca2 plus channel will induce the increased flux of Ca2 plus inside the cell, right? Once Ca2 plus is being increased inside the cell, then the primary focus here would be the translation of this POMC set of genes. And once this POMC set of genes are being translated, then further they are being carried out to the further processes to switch on the insulin to carry out the changes. And we will talk about the ATP inducement or appetite inducement or the appetites, you can say suppression inside the cell due to this insulin molecule only. Once over, the blood vessels would be having a high flux of glucose molecule inside the cell, right? So let's see those processes also. Okay, so now once the polypeptide is being created, that is the insulin. Insulin is a form of pre-proinsulin, right? Which form does insulin exist? Primarily after the translation has been created is pre-proinsulin. Once the translation is being carried out, this pre-proinsulin is further modified to the proinsulin by the molecular scissors that we call as proteases. Proteases does what? There is a amine group and there is a carboxylic group, right? Now these two groups contains the information that is the signaling peptide, that the peptide is being carried out to the molecular processes or by the molecular scissors. What does this protease does is that it just degrade this sort of amine and COO terminal where the signaling peptide is, and the peptide sequence is being cleaved out. And once this peptide sequence is cleaved out, it is further carried out to the next further process, downstream process, that is proinsulin generation and proinsulin generation, right? Now this proinsulin is not further activated insulin, but it is a sort of second step to the activation of the insulin. And this proinsulin is being stored in the vesicles, in the pancreas, so that further whensoever there will be the higher flux of this insulin molecule, for the glucose molecule, then the signaling cascade will induce the molecular seizure to cleave this C domain of this polypeptide chain. If you can see here only, there are three domains A, B and C, where A, B are together by the disulfide bond, that is the cysteine bond, and this C1, this C peptide is being, you can say, away from the sequence. Once the molecular scissors are being carried out to carry out the signaling cascade, they will cleave this C domain also and mature this insulin molecule into the activated insulin that is being known as this A domain plus B domain only, right? Once this A domain and B domain is being generated, then further phosphorylation and the methylation of certain residues of the polypeptide will induce activated insulin that is being further packed into the vesicle, and this is a sort of military tank that is having all sort of weapons into a truck that is being packed into some vesicles, and once those vesicles are being packed, then the signaling cascade are withheld for a moment. Whensoever there will be a higher flux of glucose outside the cell, then the induction is in such a way that the vesicle will generate those activated insulin molecule or give rise to those activated insulin molecule that would carry out the degradation of the glucose outside the cell, right? Now this is the primary thing. Now there's one more thing here to understand is that appetite suppression by insulin molecule. For that, we need to understand some aspects of neurosciences and some aspect of this insulin cascade, right? Okay guys, so the primary thing here to understand is that how does this appetite is being suppressed by the insulin molecule. So say somehow in pancreas the insulin is being generated due to the flux of glucose outside the cell that would signal the suppression of the appetite. How does this process happen? This is the primary thing to over here to understand, right? Now the appetite goes on with two signals and off with two signals, that is eat and do not eat. And the two of the signaling peptides that are being generated in the hypothalamus. So this is the brain, and here is the hypothalamus, and here are the two sort of pituitary glands, that is the anterior and posterior pituitary gland. And apart from that, we do not need to go to the pituitary gland in detail, because here the main focus is on the hypothalamus. Because hypothalamus induces two of the signaling neuronary signaling peptides, that is neuropeptide Y and MSH, and melano-stimulating hormone, right? These are melanocyte-stimulating hormone. Now these are the two sort of peptides that are being induced in the hypothalamus once the signaling is being carried out. And the cell is articulate cell that has two sort of neuronary peptides to be induced whenever there is a flux of glucose outside the cell. You can see the diagram again and again, because I'm not going to discuss it further about the role of these hormones. So again, neuropeptide Y is an hormone that induces eat-on, means you have to eat, and MSH, this melanocyte stimulating hormone, is the particular neuronal signal that induces do not eat suppression hormone, right? Now the primary thing over here is to go for the leptin. Leptin is a peptide. Again, leptin. Leptin is another sort of hormone that has been generated for this inducement of the appetite, and we will not focus here solely on the leptin, but the crosstalk is very important to discuss primarily here only. We will discuss in another sort of video what is leptin in detail and how does it induces both the sort of suppression and activation of the appetite, right? For now, we have the insulin molecule, and once insulin molecule is bound to this NPY molecule, so leptin, if once bound to this NPY, it would not activate it. Look, leptin is a polypeptide that activates the neuronal signals, that is NPY and MSH alpha, right? But we will not focus solely on the leptin, because insulin once bound, no further signal would cause this switch on to the appetite, right? And once leptin is bound to this MSH alpha, it would induce that do not eat. So these are the sort of processes which insulin is bound to, then leptin cannot do anything. Leptin cannot induce the appetite anyways. Now, these are the sort of ways in which this insulin induces the appetite also, and insulin always triggers the signal of do not eat. And whensoever there is the high flux of the glucose outside the cell, insulin will generate a primary signal is that do not eat, you do not want to eat. Your brain is quite stable right now, it is having a quite enough amount of glucose. You do not want to give your body enough glucose since it would be generated into the TAGs, that is triacylglycerol. So the adipose tissue, that's bad if the number of adipose tissue increases in the body, that would induce several sort of cancers, right? Now this is the whole process in the way the insulin is being generated, and it carries out the signaling. Now the insulin carries out two kinds of signaling. Right, primarily is degradation. Let me speak it, right? The primary is the degradation of the glucose into the ATP molecule, conveying the signal for glycolysis, citric acid cycle to the oxidative phosphorylation. Second primary signaling is that if not, because it is antagonistic to what? Glucose. So what it will do is it would increase the generation of glycogen molecules, or it would increase the generation of the TAGs, so that the glucose flux is being diminished into short packages that could further be broken down in the emergency condition, where glucagon, the alpha cell that stimulates the glucagon, would stimulate this high amount of polymers to be broken down into this mono unit, and further insulin will degrade it into its ATP units, right? Now these are the sort of things that I need to discuss about the insulin process. How does insulin work in the body? Hope guys that you all like this video. If you like this video, keep on subscribing, and thank you.